Hello everyone. For TYBSc in Fermentation Technology, today we are going to start our third chapter, which is sterilization. And in today's lecture, we are going to study the different methods used for the purpose of sterilization. Now, in this chapter, we are going to learn, or in this topic, we are going to learn the different contents, means what is sterilization, then history of sterilization, which different types of methods are, or which different methods are used for the sterilization purposes. First of all, as we all know, what is sterilization? Sterilization is nothing but the process used to remove or destroy the viable forms of microorganism, or the sterilization can be defined as a process by which all the viable forms of microorganisms are removed or destroyed. And you might be knowing that microorganisms are present everywhere, and it can cause infections or different types of diseases, and it can also contaminate the different types of products. So it is very important to remove or destroy such microorganisms so that our product will remain safe for the longer use. This sterilization technique was first used by the scientist Louis Pasteur. He developed steam sterilization in the then hot air oven and autoclave, and also introduced the method of pasteurization to preserve the milk from the spoilage. And later on, Joseph Lister applied this technique for the antiseptic technique in the surgeries. Then we will move towards the actual point, which is methods of sterilization. Three different types of methods are used for the purpose of sterilization, which is physical method, second one is chemical method, and third is filtration method or the mechanical method. First we will see the physical method. In physical method, 4 different types of sub-methods are used, which is sunlight, second one is drying, third is heat. In heat, we can use 2 types of heats, which one is moist heat and second one is dry heat, and fourth important type is radiation. We'll see one by one. First one is sunlight. Now it is responsible for the spontaneous generation in the natural condition. In natural condition, as different types of ultraviolet rays and heat is released from the sun, these sunlight can be used to kill the bacteria or the harmful microorganisms present inside the water or in the natural environment, so that it will be safer for the use. For example, there are different types of tanks or lakes are present in the open areas. Due to the sunlight, ultraviolet rays released from the sun and heat generated from the sun, the microorganisms present in the these open lakes can get killed and water quality can be improved. Next one is drying. Now, if the food content or any material contains moisture, you might be knowing that for the growth of microorganism, they requires adequate amount of water. And if this adequate amount of water is present or moisture is present inside any foodstuff or any substance, then the chances of contamination is more. So to prevent these substances, the drying process is generally used, and this drying in the air has the dangerous or strong effect against the harmful bacteria. but generally the spores are not affected by drying technique. Now you might have seen that grains are there, like wheats and bajras. The drying process is carried out in such grains so that the moisture content in grains will decrease and they will remain in proper condition for the longer time. Then the third most important physical method is heat sterilization. Now in heat sterilization, it has two types. We have already seen that in heat sterilization there are two types of heat sterilization. One is dry heat sterilization and second one is moist heat sterilization. Then in dry heat sterilization, dry heat is used for the sterilization of different materials, and these materials are heated in the air or fire to kill the or to remove the harmful microorganisms, and even the most resistant spores which were not killed in the drying method can also be killed in the dry heat sterilization method. Now in this dry heat sterilization method we can use different types of equipments. For example, hot air oven can be used. Then red hot sterilization can be performed, flaming and incineration. Now in hot air oven, hot air ovens are the electrical devices which use dry heat sterilize. We can increase the temperature of hot air oven from 50 to 300 degrees Celsius by using thermostat. Now thermostat is used to control the temperature in the different devices, and by using this thermostat we can control the hot air oven temperature, and this hot air is generally spread in every part of oven by using air circulating fan inside the हॉट एयर ओवन. आप सब लोगों ने हॉट एयर ओवन देखा होगा, उसमें हॉट एयर जनरेट की जाती है एंड थर्मोस्टेट यूज़ करके उस हॉट एयर ओवन का टेम्परेचर रेगुलेट किया जाता है. एंड डिफरेंट टाइप्स ऑफ फैंस आर सेट टू सर्कुलेट दिस हॉट एयर ओवन इन एवरी पार्ट ऑफ द ओवन. So simply by increasing the temperature of oven we can remove the harmful microorganisms or each and every type of microorganisms and even spores from the different types of equipments or glasswares. Then second type of dry heat sterilization is red hot sterilization. Now while performing the different types of practicals, we use inoculating needles or bacteriological loops, then straight wires or forceps or spatulas. These different materials, generally what we do, we directly keep this inoculating loop on the Bunsen burner. आप क्या करते हो, बंसन बर्नर पर अपनी लूप जो होती है वो होल्ड करते हो टिल इट गेट्स कंप्लीटली रेड हॉट. व्हेन इट गेट्स कंप्लीट रेड हॉट, द all the types of microorganism present on the loop gets killed and the loop gets sterilized. So heating any material till it gets red hot and all the types of microorganisms present on it gets killed. This technique is called as red hot sterilization. And then flaming technique. Now flaming technique generally यहाँ क्या किया जाता है, directly the material which we have to sterilize is passed through the flame. फ्लेम से हमें क्या करना पड़ता है, हमें जो भी मटेरियल स्टरलाइज करना है उसे सिर्फ पास करना. बट these while passing through the flame, it is not necessary to hold it till it gets red. रेडनेस आने तक होल्ड नहीं करना है, सिर्फ हमें क्या करना है, फ्लेम के ऊपर से उसे मूव करना है. एंड डिफरेंट टाइप्स ऑफ स्कैपल्स आर देयर, then glasswares are there, glass slides are there, cover slips are there. To sterilize such materials or glasswares, flaming technique is used. Then fourth type is incineration. Now incineration is nothing but waste treatment process which involves the organic substances contained in the waste material. जो भी वेस्ट मटेरियल में ऑर्गेनिक सब्सटेंसेस प्रेजेंट होंगे, टू रिमूव और टू ट्रीट सच वेस्ट मटेरियल, द टेक्निक यूज्ड for the treatment of such waste material which contains organic materials are known as incineration. And these method is used to sterilize generally the medical and other biohazardous waste before discarding. कोई भी मेडिकल वेस्ट होगा या फिर बायोहजार्डस वेस्ट होगा, उसे डिस्कार्ड करने से पहले इट इज़ वेरी इंपॉर्टेंट टू परफॉर्म इंसिनरेशन प्रोसेस ऑन इट. नाउ दिस वर ऑल अबाउट ड्राई हीट sterilization. In dry heat sterilization, we have seen the use of hot air oven, then red hot sterilization, flaming and incineration. Now the second type is moist heat sterilization. Generally moist heat sterilization technique is used for the most sensitive materials and the materials through which steam is permeable. Generally your culture medias are there, then different types of substances are there in which it is very important to sterilize those substances, but the quality of that product should also not be destroyed. So moist heat sterilization is used. Now in these moist heat sterilization also has three types. Supplying heat depending on the temperature used for the sterilization of material. Moist heat sterilization has three types. First one is below 100 degrees Celsius, second one is above 100 degrees Celsius, and third one is at 100 degrees Celsius. मतलब substances we have to sterilize below 100 degrees Celsius, in which we can use pasteurization technique. Now you all might be knowing that pasteurization technique is generally used to reduce the bacterial population present in the liquids such as milk, and it destroys the organism that may cause the spoilage of that milk. मिल्क का स्पॉइलेज प्रिवेंट करने के लिए या फिर स्टॉप करने के लिए हम क्या करते हैं, मिल्क को एट 72 डिग्री सेल्सियस वी बॉईल द मिल्क एंड सडन कूलिंग इज परफॉर्म्ड ऑन दैट मिल्क so that the milk will remain safe or good quality for the use. So pasteurization technique is generally used to prevent or sterilize the different substances, but due in pasteurization spores are not affected or they remain unaffected in the pasteurization technique. Second technique in below 100 degrees Celsius is incipation, means heating at 80 to 85 degrees Celsius for half an hour daily on थ्राइस कॉन्जिक्यूटिव डेज जो भी सीरम्स होंगे या फिर एग मीडिया होगा, these egg media and serums are generally heated at 80 to 85 degrees Celsius for half hour. हाफ आवर के लिए 80 to 85 डिग्री सेल्सियस पर हीट करना, एंड दीज heating is performed for the continuous 3 days, and after 3 days our serum or egg media gets sterilized, and this whole technique is called as insipisation. मतलब यहाँ क्या किया जाता है, हर दिन हमारा जो भी सीरम होगा या फिर एक मीडिया होगा, उस सीरम को 80 to 85 डिग्री सेल्सियस की 80 to 85 डिग्री सेल्सियस टेम्परेचर पर हाफ आवर के लिए हीट किया जाता है. When this heating is completed, then on next day same process is performed for next 3 days. After 3 days the content get properly sterilized. This was about below 100 degrees Celsius. Then second one is at 100 degrees Celsius. Now name itself indicates that the substance is directly boiled at 100 degrees Celsius. Now boiling is very simple method for water disinfection. हम घर में भी क्या करते हैं, वाटर बॉईल करके रखते हैं, सो फॉर द ड्रिंकिंग पर्पज और फॉर various household uses. What happens due to boiling, the microorganisms or the bacteria whichever present in the water gets killed and water becomes safe for the consumption. Heating water at high temperature or 100 degrees Celsius kills most of the pathogenic organisms and particularly viruses and bacteria which causes waterborne diseases. So this technique is very effective. Water is boiled for at least 20 minutes so that it can be safer for our consumption. And second type is sterilization. In FI you might have already learned about the sterilization process. In the sterilization process, the exposure at 100 degrees Celsius for 20 to 45 minutes for 3 successive days can completely sterilize the different types of sugars, gelatins and serum containing media. यहाँ क्या किया जाता है, फॉर 20 to 45 मिनट्स के लिए जो भी हमारा शुगर्स होंगे या फिर जिलेटिन्स होंगे उनको 100 डिग्री सेल्सियस पर एक्सपोज किया जाता है सो दैट दे गेट्स कम्प्लीटली स्टेरिलाइज्ड. एंड द थर्ड टाइप in moist heat sterilization is above 100 degrees Celsius. Now above 100 degrees Celsius you all might have used autoclave, and this autoclaving process is most reliable process for the purpose of sterilization. Generally in microbiology lab the autoclave is most important equipment for the process of sterilization. Now in autoclave the pressurized steam is used to destroy the microorganisms. Generally different types of glasswares are there, medias are there, then reagents are there. These all the materials are heated to the 121 degrees Celsius for 15 to 20 minutes under 15 lbs pressure. When the temperature rises to 121 degrees Celsius at 15 lbs pressure and it is hold for 15 minutes, all the types of microorganisms, even the spores present inside those materials, then it may be medias or reagents, gets completely killed, and these media gets 100% sterilized. Then fourth and most important type of physical method is radiation sterilization. Now, in radiation sterilization, again it has two types. First one is non-ionizing or hot sterilization. In non-ionizing sterilization technique, first one is infrared rays. Now these infrared rays are generally used for the sterilization of medical equipments like syringes or catheters. Now catheters are the thin tubes which are generally used to remove the liquid body fluids from the patient's body. And by using infrared rays, rapid sterilization can be performed of the different types of syringes and catheters before they are packed and used for the patients. अगर ये syringes होंगे या फिर catheters होंगे, if they are not properly sterilized, then they may cause infection to the patients. So it is very important to sterilize all these materials, and for the sterilization of these materials, infrared rays are generally used. Then next one is UV rays. UV rays are used for the disinfecting enclosed areas such as entryways or operation theaters and labs. जनरली जो भी ऑपरेशन थिएटर्स होंगे या फिर लैब्स होते हैं या फिर कोई भी क्लोज्ड एरिया होगा या फिर डोर एंट्री का जो एरिया होता है, टू स्टरलाइज सच एरियाज जनरली द यूवी रेज आर यूज्ड. And the second type is ionizing or cold sterilization. For the ionizing sterilization, gamma rays or X-rays are generally used, and these gamma rays or X-rays are used for the sterilization of plastics, syringes, different swabs, then animal feeds, oils, grease, fabrics or metal foils. ये जो भी नॉन लिविंग थिंग्स है, टू स्टरलाइज सच material, generally the gamma rays or X-rays are used. Now there are different types of advantages of the radiation sterilization, that it is properly clean and dry process, and it ensures that our whole objects get properly exposed and it gets completely sterilized. जो भी मटेरियल हमें स्टरलाइजेशन करना है वो प्रॉपरली एक्सपोज होके वो मटेरियल प्रॉपरली हर एरिया से स्टरलाइज होगा. इट इज़ प्रॉपरली परफॉर्म इन द रेडिएशन टेक्निक, बट इट हैज़ few disadvantages, that there is harmful effect on the humans, or there is a threat of harmful effects caused on the human if they are exposed to these rays for the longer time. अगर कोई भी ह्यूमन बींग्स इस रेडिएशन को एक्सपोज होता है या फिर ये प्रोसेस ज़्यादा लेंदी हो जाती है, तब it may cause harmful effects, and it requires very well qualified persons for the using these radiations for sterilization purposes. These were all about the physical methods used in the sterilization purpose. Then in next lecture or in next video we will study about the chemical methods used for the sterilization process.